Hello, I'm Kenneth Holmes, and today I'll be talking about the pathway of sound transduction. As sound waves travel, they are compressing and decompressing the air around us. Utilizing the shape of the outer ear, they are funneled into the ear canal. This triggers the tympanic membrane to vibrate specialized bone within the middle of the ear. These bones then move to vibrate an oval window, leading to the cochlea in the inner ear. The cochlea converts the mechanical movement created by the vibrating of the tympanic membrane to moving the specialized bones to vibrating the oval window, which vibrates fluid inside the cochlea, into action potentials that can be understood by the brain. The outer ear, also known as the auricle or the pinna, is the visible portion of the ear. It is mostly made up of cartilage, and its shape helps move sound towards the ear canal. As they enter the ear canal, they will approach the middle ear. The middle ear is defined as the space between the tympanic membrane and the fenestra vestibuli. It starts with the tympanic membrane that connects the three specialized bones. These bones make up the ossicles. The ossicles will move and vibrate based on the movement of the tympanic membrane. The third bone of the ossicles then transfers that motion to the fenestra vestibuli. The tympanic membrane, otherwise known as the eardrum, is the boundary for the outer ear and the middle ear. As compressed air hits the eardrum, it causes it to flex. This will make it compress and decompress itself. This motion is then transferred directly to the ossicles. The ossicles are made up of three bones that evolved from the jawbone of our reptilian ancestry. The bones are the malleus, or the hammer, the incus, or the anvil, and the stapes, or the stirrup. The malleus is moved directly from the tympanic membrane, and that motion is sent to the incus. The stapes then moves in conjunction with the motion delivered by the incus. Each motion increases the movement made from the one before, causing the actions to be multiplied by up to 100 times that of the tympanic membrane. This is so the vibrations can be transferred to the fluids located on the other side of the fenestra vestibuli. The fenestra vestibuli is also referred to as the oval window. This is a membrane that sits at the opening between the cochlea and the middle ear. The vibrations of the stapes cause the oval window to compress and decompress, sending waves through the fluids within the cochlea. The inner ear consists of the cochlea and the vestibular apparatus. The vestibular apparatus are specialized fluid-filled tubes that are laid out in a way that can help sense different motions of the head. There is a tube for forward and backward motion, side to side motion, and tilt left and tilt right. They can also help us sense acceleration. All of this is to assist us with balancing. The cochlea is responsible for converting the vibrations that are sent from the tympanic membrane to the ossicles that move the oval window into sound signals sent via action potential. The cochlea's spiral shape creates a cone-like bone that is known as the modulus. Slicing into the cochlea, you can see that is a spiral around the modulus with a hollow opening with segments of three parts. The scala vestibuli makes up the top segment. The scala media makes up the middle segment, and the scala tympani is the bottommost segment. The scala vestibuli and the scala tympani share one specialized fluid, while the scala media has a different specialized fluid. All of this assists with the conversion of mechanical actions into action potentials. The scala vestibuli, the uppermost cavity in this picture, is filled with perilymph. This is a fluid that is high in sodium and low in potassium. To maintain this fluid, the scala vestibuli is separated from the scala media by the Reisner membrane, also known as the vestibular membrane. The oval window is directly connected to the scala vestibuli. The scala media contains endolymph. This fluid is high in potassium and low in sodium, the opposite of perilymph. This potassium is important for the actions that take place in the scala media. The vibrations delivered through the scala vestibuli will transfer to the scala media. This vibration is moved to the basilar membrane, which is located between the scala media and the scala tympani. The basilar membrane is a structure that is more rigid near the base and more elastic near the apex. This allows for the separation of frequencies. Higher frequencies will flex and move the more rigid base, transferring the energy early on. Lower frequencies will continue to move throughout the basilar membrane, transferring energy throughout the basilar membrane. And the lowest of frequencies in our hearing affect the apex more so in the basilar membrane. All of these movements are transferred to the organ of Corti, which holds specialized hair cells. Hair cells are named as such because they have specialized cilia that are raised off the cell. There is a primary hair known as the kinocilium and secondary hairs that are known as the stereocilia. Each of the cilia are connected by a protein known as tip link. As the basilar membrane vibrates, endolymph moves into the area of the hair cells. This motion causes hair cells to brush against the tectorial membrane. As the stereocilia flexes towards the kinocilium, this opens gates to allow potassium that's within the endolymph to enter the hair cells. As more potassium enters, the cell becomes depolarized. This opens gates that allow calcium to flow and which releases glutamate to a local synapse. This is the beginning of our action potential, sending the signal for us to hear sound. As the stereocilia moves away from the kinocilium, the cell will close the gates and begin to hyperpolarize.